Yesterday we talked about active transport of small molecules, of ions and things like that. Today we're going to talk about active transport of large molecules and more than a small amount of molecules, so larger quantities of molecules. And so the term for that is called bulk transport. And bulk transport is always going to involve the use of vesicles, and so it's always going to require energy, so it's a type of active transport. So it's active transport of large amounts of molecules using vesicles. Okay, and it can be divided into two main categories. There's going out of the cell and going into the cell. So we'll talk about going into the cell first. If it's going into the cell, it's going to be called endocytosis. Okay, endocytosis is the creation of a vesicle inside the cell that is going to bring molecules from the outside to the inside. Bringing molecules from the outside to the inside. By creating a vesicle inside the cell. So if you're going to draw endocytosis, what you would see is phospholipid bilayer, something like that. You get a protein on the outside that the cell wants to eat, right? Then you have the indentation forming in the phospholipid bilayer. That indentation pulls in whatever that protein was. Okay, then it seals off. And it seals off, holding that protein on the inside. And then the final step is you've got a phospholipid bilayer, you've got a vesicle inside the cell that contains whatever thing on the outside it wanted to take in. Okay? It's the basic gist of endocytosis. Alright? There are three different types of endocytosis. The type that we've drawn here, if it's taking in a protein, that type would be called phagocytosis, which literally translated means cell eating. Okay, and basically what that means is it's going to take in a large macromolecule or a group of large macromolecules and then break them down and use them for food. Right, so taking in large macromolecules and breaking them down to use their monomers. Okay, so how does this happen? How does a macromolecule get broken down into monomers? Yeah. Lysosome. So a lysosome would attach to the vesicle that's created through phagocytosis. And inject hydrolytic enzymes And how do the lysosomes know to attach to that specific vesicle? Cell signaling proteins. And what do cell signaling proteins attach to? What are they looking for? So the cell signaling proteins are like the receptors, but what are the receptors actually looking for? Glycoproteins and glycolipids. Mark a vesicle for digestion. Okay? Starting to see some connections here between things. All right. Let's talk now about the next type. The next type is called penocytosis. Which again, literally translated, just means cell drinking. And this means that a vesicle is going to be created that contains just a solution that contains water and dissolved solutes. Do you think that a lysosome is required for this? Does a lysosome have to come in and break down anything in that? No, it's just dissolved solutes, so they don't need to be broken down any further. Okay. Last type is called receptor mediated. Endocytosis. In receptor-mediated endocytosis, there's a receptor protein that's going to look for a specific type of dissolved solute, like let's say glucose or something like that. And if there's glucose present, then that triggers the creation of a vesicle. Okay, so specific receptors. Detect molecules in solution. And trigger the formation of a vesicle. Okay, so in the book it mentions that receptor-mediated endocytosis is more efficient than penocytosis is, and the reason for that is that receptor-mediated endocytosis is looking for a specific type of solution that it's going to bring in. Okay, whereas penocytosis is just like doing the mystery grab bag thing, where it's like, all right, let's bring in a bunch of stuff and hope that what we need is in there, right? Receptor-mediated is just looking for one specific thing, and if it finds it, then it brings it into the cell. Okay? So the other type is called exocytosis. I don't know if I've mentioned this yet. It might help you. You're seeing this word a lot. This part of the word, cyto, right? Cytoskeleton, cytosis, endocytosis, exocytosis, penocytosis. Cyto just means cell. So if it says cyto in it, it means part of the cell or something to do with the cell. This is specifically saying exocyto, right? So that means that the cell is moving something out of it, exo, right? And then this cis part at the end, that just means process. So this is the process of moving something out of a cell. And that's really all it is, right? This is the transport of molecules outside of a cell, or outside the cell, by fusing a vesicle with the plasma membrane. So what this would look like, you've got plasma membrane here, here's a vesicle on the inside. This vesicle contains a protein of some kind. It's going to then fuse with the membrane. Like this. And the membrane is going to break apart. To allow that thing out. And then you're left with, at the end, plasma membrane and the thing on the outside floating around. Okay? You'll notice how it's not in a vesicle when it's outside of the cell, because there's nothing to carry that vesicle. There's no kinesins out there that would carry that vesicle along, so it would be worthless to have a vesicle outside the cell. Right? Anybody have questions on bulk transport? Okay, so now we're going to talk about two different situations where cells can be. Okay, so we're talking about larger now. We're getting bigger picture. We talked about organelles, then we talked about cells, and now we're going to talk about how cells actually interact with one another. Okay? And so we're going to call this extracellular space. Okay? And there's really two types of extracellular space. There's what's called the extracellular matrix, right? And the extracellular matrix is really something that we only see in animals. Okay, so this is it's sometimes abbreviated ECM. And what it is a series of proteins that gives animals structure and holds cells together. Okay, so this concept is sometimes foreign to people because if you look at like a plant is like a big mass of cells, right? There's really no spot in a plant that something is getting done that doesn't have cells there, okay? But humans and all animals, in fact, are different than that. Humans and animals, we have this stuff called extracellular matrix, and that means that our cells are sometimes far away from each other. And then there is a connection in between the cells made of protein that holds them in their place. Okay? So for instance, if you look at like a bone has some cells in it that are called osteocytes. But the bone is not made of mostly osteocytes. The bone is made of some osteocytes and a lot of extracellular matrix. Right? Blood. Is blood pure cells? Now, it's a lot of water, right? And there's some cells floating around in it. And there's some proteins floating around in it too. So this is a concept that we call connective tissue, saying like, hey, not all the cells have to be directly butted up against each other to actually get something done in a human. And that's where the extracellular matrix comes into place. So to talk about the extracellular matrix, we need to understand a few different parts of it. Okay, there are these proteins, these transmembrane proteins that are called integrins. And the job of an integrin is to, first of all, be transmembrane. So it goes all the way through the plasma membrane. And on the inside, it's going to attach to that mesh of microfilaments that we were talking about earlier, the one that resists the crushing force, right? So these are transmembrane proteins that attach on the inside of the cell. To the microfilament mesh. Okay? Well, if they're attached to the microfilament mesh on the inside, what are they attached to on the outside? Well, there's another protein that's called fibronectin. And fibronectin is the protein that it attaches to on the outside. So this fibronectin is attached to the outside of an integrin. And anchors it to the extracellular matrix. Okay, so that's how the cell is attached to the extracellular matrix. Now, what is the extracellular matrix made out of? Well, it's made out of a class of proteins that are called proteoglycans. There are a lot of different types of proteoglycans. Proteoglycans can be classified as connective tissue. There are some proteoglycans that are grouped in with intermediate filaments, right, of the cytoskeleton. And so we say these proteoglycans are just a class of protein that makes the structure of the extracellular matrix. Okay, so let's do a drawing. Okay, we'll do a cell here, and another cell right here. Okay, and the cells are not touching each other, right? And so these cells need to be anchored in place so that they stay there and maintain their structure. And so the way that they do that is inside of their plasma membrane, they have transmembrane proteins called integrins. And these integrins have to have some structure that holds them in place. They don't want to just get pulled apart. So the structure that holds them in place is the mesh of microfilaments that makes up the inside of the cell. Something like that, I don't know. So here's your mesh of microfilaments making up the inside of that cell. Mesh of microfilaments making up the inside of this cell. And then there's the proteoglycans. The proteoglycans are going to be a bunch of bundled up fibers on the outside of the cells, like this. We'll label them all in a second. And then do you have any more colors available? Oh, I'm gonna have to go a fifth color. Sorry. All right. So then this is going to attach here. These are our fibronectins, and the fibronectins are the things that are attached to the integrins on the outside, and they attach the integrin to the extracellular matrix. Okay, so we got some different parts in here. These things, those were the integrins. Those things That was microfilaments. The green things, the green branchy things, those were the proteoglycans. And then the pink things are fibronectins. So, let's talk about bulk transport here. Let's say cell 1, the cell that's on the left, needs to transport a protein or needs to signal cell 2 that something is going to happen or something is happening around there. Maybe it's got an infection or something and needs to tell cell 2 about it. Okay? How does it go about signaling cell 2? Cell 1 needs to tell cell 2 something's up. Yeah? But they never touch each other, right? So the glycoproteins and glycolipids of these two cells are never going to touch each other. So how can they talk then? Yeah. Nope. Okay. Nope. Can't go straight through the proteoglycans. This is just a space in here, okay? So, like, if I wanted to throw somebody a note, right? I'm just going to, like, chuck it out there and, like, hopefully it gets to one of you, right? Here you go, Annie, and I chuck it over there, right? Yeah? Okay. So, the cellular equivalent of that is exocytosis of a protein, right? So, a protein, a signaling protein of some kind gets created in this cell, and then it gets sent out through exocytosis, and now it's just floating around in this extracellular space here. And might then get over to here where it could be picked up by a signaling protein or a receptor protein that's on the outside rather, and then that could create a second messenger inside the cell, or it could take it into endocytosis or something like that. But it tells the cells what's happening around them, right? Make sense? Okay. So that's kind of what has to happen in an animal cell because the cells are not always super close together. But in the event that cells are super close together, there's a better way to do this, okay? And so we are going to look now at cellular junctions. Cellular junctions are exactly what it sounds like. These are proteins that hold cells together, proteins that connect cells. And the most common type of junction is called an adhesion junction. Depending on which book you read, you may see adhesion junctions used as a generic term, or you may see them used as a specific term. I prefer them more as a generic term. Okay, so adhesion junctions are just specific proteins that will adhere or hold two cells together. Now you might look at the difference between cellular junction there and adhesion junction and say, well, what's the difference? Well, in a cellular junction, you're not necessarily going to hold them together. There's not necessarily any holding strength involved in that. They're just connecting the cells so that maybe they could exchange nutrients or exchange water or something like that. Okay? So let's talk about the different types of adhesion junctions. The strongest type of adhesion junction is called a desmosome. And a desmosome is the type of adhesion junction that not only connects in a very strong fashion, but it also is going to allow for a little bit of elasticity, right? So it allows for them to stretch a little bit. So this is a connection that provides a strong hold and the ability to stretch without tearing. Okay, so desmosomes are attached through proteins that are going through the plasma membrane that are attached to the cytoskeleton of each cell, and then they kind of have like a little handshake that's very similar to the way that a fibronectin works, but they connect directly from cell to cell. Okay. The problem with this is that even though they're very strong, so like for instance, the ability for my skin when I do this to not tear apart is the result of desmosomes holding my skin cells together, right? The problem is that they're not watertight though, because they're connected by these little handshaking proteins like this, and so if I didn't have something else that was holding together my skin cells, I would be leaking fluid all the time out of my skin, and that would be not good, right? So there's a second type here called a tight junction. And tight junctions are watertight. Okay, they hold the cells very close together. So that means that skin cells must have a combination then of desmosomes and tight junctions, because if I just had tight junctions and I did this, my skin would tear apart, and that wouldn't be good. But if I just had tight junctions, or sorry, if I just had desmosomes rather, I could do this and my skin wouldn't tear apart, but I would leak all the time, right? So I've got to have a combination of tight junctions and desmosomes in order to get this nice watertight, non-tearing skin that I have all over me, right? Okay. What about if cells just need to talk to each other, right? So they don't need to necessarily attach here. They don't need an adhesion junction. They just need a communication junction of some kind, right? So the two types of communication junctions are gap junctions, which are found in animals, and plasmodesmata that are found in plant cells. And the purpose of gap junctions in plasmodesmata is to allow for the transfer of ions and water and nutrients between different cells. Ions, water, and nutrients directly between cells. So if I had some epithelial cells here, okay, and I wanted them to be held together very tightly, they might get held together tightly by some desmosomes. That's not correct. Here's a desmosome. Here's a desmosome. Here's a desmosome. Here's a desmosome. And then in between the desmosomes, they are held together by fibers. Okay, so fibers hold them together like that. Okay, but you can still tell that even though these are holding them together, and those fibers on the inside will give them a little bit of stretch, there's still some places where water could move through here. Okay, so they are going to require some other type of thing to be watertight, and the other type of thing is a tight junction. And a tight junction is when the proteins actually go all the way across like this, and they sort of stitch together the cell in order to block any water from being able to travel through there. Okay, so these things here that are desmosomes, and these are tight junctions. Okay. Then you can also have channel proteins on either side. So imagine down here I want to have another cell and I want to communicate from one cell to the next. So I have some channel proteins. And here's another cell here. And I want these two cells to be able to directly communicate with one another. So the channel proteins of them, they butt up against each other like this. And this would be called a gap junction. Because that would allow for stuff to go from one cell through this gap junction into the other cell. So again, you can do the same thing over here. Desmosome, 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 desmosome. And then they're connected together by proteins, by intracellular proteins rather. And then you've got the stitching together of the cells by a tight junction. Questions on that?